student at the University of Washington. We are all trained in really specific techniques in a specific field, and we kind of live in these little bubbles. And the Engage program has provided us with this unique opportunity to get a different type of training that we don't usually get as graduate students. So we've learned how to talk about our science in a way that's engaging, enlightening, and informative to audiences that we often don't interact with. And you can learn about this program if you're interested at engage-science.com, or you can also Google it, Engage UW. And now we're going to introduce our second guest. So Siva Kasinathan is pursuing both a medical degree and a PhD, so he'll be a double doctor, at the University of Washington and the Fred Hutchinson Cancer Research Center. He's interested in genetics, especially in the context of disease. So today, Siva will tell us about mapping largely unexplored part of our genomes. Let's welcome Siva to the stage.
Okay, can you hear me?
Yes.
All right. Thank you, Emma, for the introduction. So I'll be talking about parts of our genomes that we haven't really been able to explore very well. But before jumping into the genetics or the genome, I'd like to talk a little bit about this work of art, since Anne did such a nice job of introducing this idea of a genome. So this is a work of art by Grant Wood, who's a painter from Iowa, not too far from where I went to college and where I grew up. And this is called Dinner for Threshers. A thresher is someone who separates the seed from the rest of a plant. And I really was drawn to this because of the richness that is portrayed in the image. So in addition to these farmers who are sitting around the table after a long day of work enjoying dinner, there is a lot of depth to the picture. So there are these textures in the wallpaper, there's even texture in the tablecloth and the shirts that some of these guys are wearing. I didn't come across this picture at an art museum or in a textbook, but instead I came across it in something like this. It was a jigsaw puzzle that I got at a county or state fair. And the puzzle I got had more than 6 pieces, but for our purposes today, we'll think about a really simple 6-piece puzzle. And here are our 6 puzzle pieces. And the reason we're talking about this puzzle is because it's really inspired a lot of the thinking that I do now as a graduate student. So I want you to close your eyes and think in your heads a little bit about how you go about putting together a jigsaw puzzle. What strategies do you use? Hopefully everyone here has had experience putting together a jigsaw puzzle.
Find the edges.
Find the edges. Okay, so that's a good suggestion. Other things that people usually throw out are things like look for patterns that are shared between puzzle pieces, kind of try and group things based on what the front of the box looks like. So you have to have some knowledge of what the picture you're trying to assemble is. But all of these are really great strategies. So we can think about 2 of these puzzle pieces that we think might go together. So here's a piece that is an edge. Here's one that is probably the bottom edge. And we think these might go together. And we can test this out. In fact, if we wanted to, we could test every possible pair of puzzle pieces since we have such a small number. And using this approach, we can construct a little map or a set of instructions that can guide the assembly of this puzzle. So here in this depiction, I'm saying that this puzzle piece is close to this one. And in this way, I can draw a set of arrows that connects all the puzzle pieces together and allows me to reconstruct the full puzzle. Okay, so the puzzle that I'm interested in putting together is obviously not a jigsaw puzzle like this, but is a puzzle that has to do with the genome. So this is pretty similar to the picture that Anne had up earlier. This is a this is something taken from a textbook. And as Anne explained, the cell is the fundamental unit of life, and we're made of trillions of these cells. And for the most part, each cell in the human body has a copy of the genome. And the genome is organized into these units, these X-shaped things called chromosomes. And if we were to uncoil a chromosome all the way down, we would find the DNA double helix that was introduced earlier that's made up of these 4 letters. This is a picture from a textbook. Do chromosomes really look like this? It turns out they do. Here are the chromosomes from a single human cell, in this case from a human male. There are 23 pairs of chromosomes that are numbered 1 through 22, and then there's an X and a Y chromosome. And each of these chromosomes, again, if we were to zoom in, is composed of this linear sequence of A's, C's, G's, and T's. Looking at chromosomes this way actually turns out to be relatively useful. There are a number of human diseases that are caused by gain or loss of entire chromosomes. Down syndrome, for example, is caused by an extra copy of chromosome 21. So if we were to look at an individual who doesn't have Down syndrome, we'd find 2 copies of chromosome 21. But an individual who has Down syndrome would have 3 copies of chromosome 21. Now, it's not just Down syndrome that is characterized by gains or losses of entire chromosomes. There are a number of other disorders as well. Since I work at the Fred Hutch Cancer Research Center, I think about cancer every now and then. And it turns out that virtually all cancers are associated with chromosomal abnormalities that can include gain or loss of chromosomes, entire chromosomes, or chunks of chromosomes. So since chromosomes and the number of chromosomes that cells have is so important, you might imagine that there are mechanisms in place that help cells ensure that they have the proper numbers of chromosomes. And one of those is called the centromere, which is a spot on each chromosome that ensures that cells get the correct number of chromosomes at each cell division. So if we were to look at a single chromosome, you'd find the centromere at this part where the chromosome sort of pinches in on itself. And if the centromere is involved in any of these diseases like cancer, then we should be able to look at the DNA sequence of the centromere and say something about in what ways it can contribute to disease. So for example, a really simple experiment might be looking at the centromeric DNA in cancer cells versus normal cells and asking if there are differences. And you might think that this is something that's really easy to do. In part because of this event that happened about 17 years ago. This is Bill Clinton announcing what is billed here as decoding the book of life. So 17 years ago, the Human Genome Project, or at least the major phase of the Human Genome Project was completed, which should, in theory, allow us to look anywhere in the human genome and know its sequence and the way that sequence relates to the rest of the genome. So why is this difficult at centromeres? So just a quick aside in terms of terminology. Instead of talking about decoding the book of life, I'll talk about genome assembly, which is the process of actually assembling the genome into a picture from the individual component DNA sequences. And we'll talk a little bit more about this process in just a second. We can do the same thing as assembling jigsaw puzzles with DNA sequence, and it turns out that the initial Human Genome Project only succeeded in piecing together somewhere between 90 and 95% of the human genome. So there's some 5 to 10% of the genome that remains unassembled, and these parts of the genome that we haven't quite pieced together include the centromeres that I'm interested in. So you can imagine how frustrating this is to be interested in part of the genome that you think has been put together by the Human Genome Project, only to find that it's in this 5 to 10% that we haven't quite pieced together. So why is it that we can't piece together this last little bit of the genome? So the reasons kind of have to do with the ways in which we sequence genomes and go about doing genome assembly. So instead of just one copy of the genome, when we go about sequencing a genome, we take the genomes from a population of cells. This usually means tens of thousands to millions of cells. And we have to randomly fragment the genomes of these cells in a process called shotgun sequencing. The reason that we have to randomly fragment the genomes before sequencing them is because we don't have technology that allows us to sequence a chromosome from end to end. So we blow up these genomes from this population of cells, and we typically end up with millions to billions of little pieces that we then need to piece together to reconstruct the picture of the genome that we want. I should also mention that in a lot of these efforts to assemble genomes, we don't have the benefit of the image that comes on the box. We don't really know what the genome is supposed to look like to begin with. So we're stuck piecing together these millions to billions of pieces just based on the kinds of things we talked about earlier. We don't really know what's an edge and what's not, but we can try and figure out which pieces go together based on the kinds of patterns they contain. So this might go something like this, right? So we can assemble what we think is this farmer's face just based on looking for shared patterns between the pieces. And this works out pretty well for a big chunk of the genome. This is at 90 to 95% that we have pieced together. But in other parts of the genome, we run into some trouble. So this might be in parts like the wallpaper in our image, where we don't really know how these pieces go together. We don't really know if they overlap or are supposed to overlap. And so we get into some trouble in trying to piece these regions together. So in the world of DNA, the farmer's face here might correspond to unique sequences where the organization of the A's, C's, G's, and T's that make up DNA is pretty distinctive, while the wallpaper might correspond to what we think of as repetitive DNA sequence. So in this case, I'm schematizing an A, T, and a C that repeats over and over again. So it's in these repetitive regions that we can't really piece the genome together. So when we go about assembling genomes, we're after something that looks like this, where we have the entire picture, but what we end up with is something that looks like this, where we're missing a lot of the detail wherever there is a repetitive pattern, like the wallpaper or the tablecloth or this guy's shirt. And so this is pretty frustrating when you're trying to study a region like the centromere, which is embedded in these repetitive regions. So we haven't really been able to get a good idea of what centromeres look like on a large scale, or how centromeres might vary between individuals, for example. So this is the problem that I kind of wanted to try and get at. And I want to go back to this idea of coming up with a set of instructions for doing the assembly, since this is really what I'm after. So here again is a really simple set of instructions for assembling this farmer's face, where the arrows between the puzzle pieces just tell us the order in which to assemble this sequence of pieces. We can do the same thing with DNA sequence, where a circle here represents a particular sequence, and an arrow between two circles just tells us that we should join those two sequences together. In repetitive regions, things get a little bit more complicated because things can be joined together in multiple different ways. And when we look at DNA, things get even worse. So this is the same kind of set of instructions as over here. Each circle represents a DNA sequence, and lines between circles tell us which sequences to join. And you can tell that this hairball isn't particularly informative in helping us assemble anything, right? If I gave you this as a set of instructions for assembling something, you could probably do it. But if I gave you this as a set of instructions, you'd probably throw your hands up and quit. I know I would. Okay. So how do we go about simplifying this, or at least rethinking this? So one thing that's helped a lot that I won't talk about today is a set of advances in sequencing technology that's effectively allowed us to make slightly bigger puzzle pieces and cheat a little bit. But the other thing that we can do is think about how we actually go about assembling the puzzle to begin with. So one way in which assembly is done now is to take this group of puzzle pieces and effectively compare every single piece against every other piece. And that, it turns out, isn't particularly effective, especially in repetitive regions, since everything looks like everything else. So what if we did something like this, where we grouped things that are similar, or that are similar based on some criterion that we can specify? So in this case, I've just grouped things based on the pattern they contain. So this grouping produces these clusters. For example, this cluster up here, these pieces are mostly brown and don't really have any other distinguishing features. The ones over here are wallpaper bits. These are bits of wallpaper with some molding, and so on. We can decompose all the patterns in this image that we're interested in into these little clusters, and then assemble things within these clusters before trying to assemble the whole puzzle. So how does this work? Well, we first, before trying to assemble human centromeres, tried to assemble the centromeres of a plant, largely because the plant has a smaller number of chromosomes than humans do, but the centromeres themselves are similar in size. So we thought this was a nice test case for our approach. Here are the clusters that we get. So in this picture, each dot represents a single DNA sequence, and dots that are grouped together in these clusters are sequences that are similar. And you can tell that we get nice groupings, and here are the assemblies that we get out. So these are these things that look like spaghetti or noodles. If we were to zoom in on one of them, each one of these is a set of instructions for assembling a particular sequence, and you can tell that this is a lot nicer than that hairball I showed you earlier. If I gave you this to assemble a DNA sequence, you could do it pretty easily, since you know where to start, and it's very easy to get from one sequence to the next just by following this set of instructions. So this was pretty encouraging to begin with. So how do human centromeres look? So we haven't quite applied this to the entire human genome yet, just small chunks of it so far. Here are what the clusters of these DNA sequences or fragments look like. You can tell that the clustering is not quite as good as what we got in plants. This is something that we're still working on, but there are clusters. Again, in this picture, each dot is a DNA sequence, and things that are close together are similar. And here are the assemblies that we get. So these assemblies, you can tell, aren't quite as nice as what we saw in plants. There's a lot of branching going on, and which means that there's some complexity in these instructions. But this is quite a bit better than what we would get using the previous approach. We might be able to figure out how to traverse these clusters and actually produce reasonable assemblies compared to this hairball from earlier. Okay, so what have I told you so far? So first, that abnormal chromosome number is something that is associated with many diseases. Centromeres are the spots on chromosomes that help to ensure that each cell gets the correct number of chromosomes. And although centromeres play this really important role, we don't really have an idea of exactly what the sequence organization at centromeres looks like, because centromeres are missing from the human genome assembly. And we've developed new tools that we think can aid in assembling these centromeres and can help us understand the biology of centromeres as well as their role in disease. So the hope is that we'll go from a picture of the genome that looks something like this, where we're missing a lot of the details that I at least care about, to a more complete picture where we have an idea of what the repeats in the genome actually look like. So with that, I'd like to thank Engage and Town Hall in particular, as well as Fred Hutch and the University of Washington the National Cancer Institute, Howard Hughes Medical Institute, as well as achievement rewards for college scientists for funding. I'll take any questions you have. Thank you for your attention.
Hi. I'm wondering if you could speak a little bit to why it was easier, why you got better clusters when you were looking at plant centromeres compared to human centromeres, and if is there less similarity in centromeres and that's why they cluster better, or so?
That's a very good question. So the question was effectively, why are the clusters better in plant centromeres and not so good with the human centromeres? And that has to do with a couple of things. So because the plant genome is a lot smaller, we can get a lot more data from plants in a single sequencing run than we can from humans. So there's just a lot more data. And with the particularities of the method, the more data you have, the more information you have to create these clusters. So that's the big reason why the clusters from plant centromeres look better than the clusters from the human centromeres. In terms of how much variation there is between or within an organism, plant centromeres, at least for this plant, are a lot more similar than human centromeres are. So the sequence variation in plant centromeres is a lot less than it is in human centromeres. So the hope is once we have our hands on a little bit more data, then the human stuff will end up looking better than the plant centromeres, but we'll see that holds true.
Hi, great talk. My question is, now that we have this new method of grouping segments based on their similarity, how long do you think it will take for us to actually complete the entire human genome? Will it be years or decades, or will we need a new method to finally complete it?
That's a good question. So the question was, how long until we complete the entire human genome? And the answer is complicated. So this method requires there to be a little bit of variation in the sequence in order to actually assemble it. So you can think about this as, in the puzzle analogy, slight differences in maybe the shading or shadows or whatever is going on in this wallpaper. So centromeres have enough of this slight difference, these shadows or artifacts from light, whatever it may be, that allow us to assemble them. But there are other repeats in the genome that don't have this variation at all. That everywhere you look, they are almost identical copies. And those repeats we're not going to be able to assemble using this kind of approach. So we'll be able to definitely assemble the centromeres using this approach. And there's still quite a bit of other repetitive stuff in the genome that we're not going to be able to assemble this way. So in order to assemble those, we'll need technologies that effectively produce much bigger puzzle pieces. And those are on the horizon. So hopefully soon.
In the little visualization slide, what do the branches and cycles mean in terms of the sequence?
Yeah. So effectively, in these depictions here, branches or cycles mean that could mean that a particular sequence or pattern is repeated over and over again, and that pattern is almost identical. So actually, the kinds of repeats that I was just mentioning that this approach wouldn't work very well with would be represented here as cycles. So which are just these closed loops, and there are some of those at centromeres as well. So this approach will allow us to assemble most of the centromeric sequence, with the exception of that sequence that is identical everywhere. All right. Thank you very much.